So, we did the nephrogenic diabetes insipidus, and it's an a B2 vasopressin receptor unable to stimulate adeno cyclase. And before I've done all that, we have the information on the rat.
A little local history.
Diabetes insipidus is not the same as sugar diabetes or diabetes mellitus. It's when the kidney is unable to function normally, so it can't concentrate the urine and conserve water like normal kidneys can. So that causes excess water consumption as well as excess water excretion and urination, as well as dilute urine. And if untreated, especially in newborns, it can cause severe dehydration and even death. There are 4 types of diabetes insipidus. There's neurogenic, gestogenic, nephrogenic, and dipsogenic. Okay, so neurogenic is the most common, and the nephrogenic is the one that is most commonly associated with the Brattleboro rat.
The neurogenic one, or NDI, is a pituitary gland that produces vasopressin. The vasopressin, the anti-diuretic, right? That's a hormone of the ADH. This is a hormone in the kidney that reduces urine output by increasing urine concentration. And NDI, it's a lack of, you can see a vasopressin being secreted. NDI is caused by the destruction of a posterior portion of the pituitary gland due to diseases and also tumors, infections, meningitis, complications, genetic defects, a whole bunch of other stuff. Desmopressin, or DDAVP, is the most common drug treatment. This one was no cure, right?
Yeah, there is one
no cure.
There's no cure. But I don't believe there's a cure for any of them. Except for gestogenic can be treated better than the rest of them, but there's no cure. It's basically lifelong treatment with DDAVP. So gestogenic is when it occurs only in pregnancy. It's also due to pituitary gland damage. Also commonly, the placenta will actually destroy the vasopressin that's secreted by the pituitary gland, so it causes the gestogenic symptoms instead. So there's treatment for DDAVP, and when infants are treated with this, the symptoms usually abate from 4 to 6 weeks after delivery, but it's likely to occur in future pregnancies and children of that mother.
The nephrogenic is one that the Brattleboro rat is most known for. It's the kidneys and sensitivities of vasopressin makes it unable to respond to the anti-diuretic effect.
And just to clarify really quick, anti-diuretic hormone, or ADH, is vasopressin. So we use that interchangeably throughout this. That's important to know.
And if it's genetic, there's no cure. And if it's drug use of disease positive, then it should with DDAVP again.
So dipsogenic is the last form. It's very similar to NDI, and it's associated heavily with excessive water consumption as well as due to thirst and excessive urination. And if the only way to tell the difference between the 2 different forms of the disease is if you start treatment and the urination decreases. So yeah, if the urination and thirst decreases, it's NDI, but if only the urination decreases but thirst persists, then it's dipsogenic. And these people are usually afflicted with water intoxication at some point in their lives because they just continue to drink water, and that's not a good idea. If you drink too much, then your blood's too thin and all that. So it's associated with all of those results. And that actually leads to a decrease in the sodium concentration in the blood plasma, and that causes all sorts of issues.
Yeah.
Yeah, that's right. Brattleboro rat was kind of a screw up. They weren't trying to breed any rats that were nephrogenic or anything. The original researchers up in Brattleboro were originally breeding rats for cardiovascular studies. And when one of the rats gave birth to 17 pups, they noticed that the bedding was constantly wet and the water always getting refilled. So after studying and doing some minor experiments and whatnot, they determined that 6 of them were drinking urine excessively. They drank about 70% of the water, which is what the picture is. It shows the size of how much water they drank a day. They treated the rats with vasopressin, and 6 of which remained. So only 6 of them remained with the symptoms. So they gave them to 2 doctors down at rat.MP4, which did further breeding of the rats. Should be on the next slide.
Yeah.
The deficient rats were then bred at rat.MP4, and then these rats were bred exclusively for nephrogenic studies, and then they were sold all over the world to use these studies.
These rats are important because they're the first experimental animal model of DI, and they were the forerunners of today's knockout experiments. They're super common today.
Nephrogenic diabetes insipidus is the actual rate that we did. Vasopressin type 2 receptors, or the V2R is how we always call it this. Is a hormone receptor coupled to effector systems by G proteins, and it functions to mediate antidiuretic activity of the ADH and the renal collecting ducts. It does its best in the GS, or the adrenal recyclase system. The defect is a recessive sex-linked gene on the X chromosome.
So there's 16 distinct V2R mutations. 6 are inactivating mutations, and the other 10 are single amino acid substitutions, where CGC goes to a CAC, which modifies the receptor, portions of the receptor that code for that accept the vasopressin, and so it inactivates the receptor basically. So Q2 mutations were examined here in this paper. The mutation was said to be at the junction of the 3rd transmembrane segment and the 2nd intracellular loop of the receptor. We have a picture of that in a minute. So the hypothesis explored in this paper is that the Q2 V2R mutant receptors might have normal ligand binding or close to that, but impaired signaling activity, so they don't in effect work. So
And this is just a diagram showing where the Q2 mutation was. They use this showing that top row is the plasma membrane transports, and the loops are the intracellular loops. So just showing that it's right before plasma membrane on this loop.
yeah, and importantly also that the one amino acid substitution occurs right here, and it causes an arginine to change to a histine.
Ligand used was today's arginine vasopressin, or AVP. PHV2R Q2 was constructed. The cDNA encoding human V2R receptor was excised and cloned radioactive mutants, antisense, oligonucleotide, neo to the single strand cDNA. The modified cDNA was excised. One end was filled with a fragment of DNA equivalence 1, then attached to the PKNH.
The COSM6 cells, I'm not entirely sure. We couldn't figure out what these were. They used all these weird different cell type names throughout this entire paper, and they kept adding new ones.
So they were using them to express the cDNA as the proteins. So they're just using, rather than studying them in a whole animal, they've just got some cells that they can get to express.
So I'm taking it that this is the most general category of cells that they were
easy to grow and are good for
That's what we thought, but we weren't entirely sure.
I'm not
So
sure
it's
their
more
origin.
specific
They
as
might.
it
I don't know if they had origin of, from kidney or anything, but.
Yeah, I don't know either. We were so confused. So they were plated and grown with those chemicals listed there, as well as those antibiotics. And they were exposed to dimethyl sulfoxide and HBSS, trypsinized, seeded, and assayed. Then they were homogenized, and the homogenate was assayed for adenylyl cyclase activity. Then they had LTK minus cells, which were also grown on plates, transfected, exposed to DNA, calcium phosphate, co-precipitate containing plasmid DNA, the plasmid that they were looking at. They treated it with glycerol and HBSS, trypsinized, diluted this, and then distributed to micro-titration wells. And then from there, the G4 maintain resistant